This is a production of Cornell University.
Over the last 20 years or so, I've been obviously doing a lot of work on photoreceptors and the phototropins, and I got kind of sidetracked, and that's what I'm going to talk about today, because we discovered that there were some interesting things going on in guard cells that, for us at least, were quite unexpected, perhaps not to others. And so we'll just continue. We're going to be talking about stomatal function, but I thought I'd give a little bit of early history. For example, way back in 1836, Charles Daubeny at Cambridge University did a very interesting experiment, and I'll show you his experimental setup in just a minute. But he showed that blue light was much better than red light in inducing plants to lose water. And to my knowledge, this is the first actual observation of a specific effect of blue light on stomatal function, although obviously he didn't know anything about that at the time. Then in 1898, Francis Darwin invented a little device called a pyrometer, and first demonstrated that light increased the rate at which you could pull air through a leaf, again indicative of some change in the physical properties of the leaf that allowed more gas movement across. It was long assumed, however, despite this experiment by Daubeny showing blue light to be so effective, it was assumed that stomatal opening was strictly responding to photosynthesis. And after all, we have chloroplasts in guard cells, so why not? And it was only much later that Ogawa showed that blue light alone doesn't have much of an effect in increasing transpiration, but against a red light background, the effect is spectacular. And you got a huge increase in transpiration. And then Zeiger and Field refined the measurements and demonstrated that it was highly specific for blue light. And as we know now, it's a response mediated by the phototropins. Well, here was Daubeny's very ingenious experiment. He used various colored glass, and he used also some solutions that were colored, and simply measured. He would put concentrated sulfuric acid, which is of course hygroscopic, takes up water, and he'd weigh it, and then he would put it under a bell jar and turn the light on. And when he turned blue light on, he got a much bigger increase in the weight of the sulfuric acid over time than if he turned the red light on. Incidentally, he also noticed something else, and that was that red light was far more effective than blue in inducing what he described as the secretion of green matter into the leaves, which of course was that he was seeing for the first time the development of chlorophyll, transformation of protochlorophylline to chlorophyllide, and then all of the phytochrome responses that follow that to get mature chloroplasts. So this was just a nice piece of work that got totally lost. This is again the first report of a blue light response, guard cell function. And then here is Francis Darwin's pyrometer. You take a leaf and then you seal a little suction cup there and just hook it up to a burette like this, and then you measure the rate at which the liquid drops. And of course, if you turn the light on, then the stomata open, the guard cells separate, and the rate at which it drops increases. And then you turn the light off, and it goes back down again. A very elegant, very simple demonstration, again, which has been pretty much lost. More recently, we identified phototropins, and one of them, Photo-1, eventually Dorello et al. Were the first to identify the second phototropin, and then Kinoshita et al. In Shimazaki's lab in Japan demonstrated that both Photo-1 and Photo-2 mediate light-activated stomatal opening. So now we've come to the point where we can actually identify the photoreceptors involved in the light response first described by Charles Daubeny. Well, in the last 3 decades or so, we've learned a tremendous amount about stomatal development. A number of different people working on this. and the genes that are responsible, the transcription factors involved. We've also, another totally unrelated in many ways, we've learned a tremendous about the ion pumps and channels that are involved in stomatal opening and closing. We learn now about the specific role of abscisic acid in closing, but there's not a whole lot of work that's been done on a possible role of the cytoskeleton in stomatal function, particularly not microtubules. There are some very nice studies from Young-Suk Lee in Korea on actin changes with stomatal opening and closing, and other papers since then, but not particularly. So let me now digress slightly, because we got interested in stomata quite indirectly. Things happen to phototropin when you turn the light on. And of course, one of the first things to happen is that you get extensive phosphorylation. Second thing, we now know that you can get binding of a 14-3-3 protein to a particular serine in PHOT2, highly specific. We know that you can get monoubiquitination here, probably other places too, according to Liscombe. And this is all work that was done by a long-term postdoc, Tongchun Seng, in the lab. So here's phototropin structure. You have a serine threonine kinase, a classic serine threonine kinase, and then you have two domains of about 100 amino acids which bind to flavin, which is the chromophore, and both phototropins have this structure. When you turn the light on saturation, these are all the different amino acids that are phosphorylated. The ones in blue were discovered by the Japanese group, the ones in black were discovered by John Christie's group, and the ones in red were ones each time you do this, you pick up different ones. We've just done another mass spec experiment, and we picked up 3 more that haven't been reported. So I think the number of phosphorylations that occurs with light saturation probably is not yet completely identified. Okay, well, one of the things that Dongchun did was a yeast 2 hybrid, and he found that PHOT2 was associating with the A1 subunit of a protein phosphatase, PP2A, and both by yeast 2 hybrid and by pull down. So then the question is, well, is that phosphatase going to play a role in, actually could show that it was the love domain that was apparently where the binding was occurring and not the kinase domain. So we hypothesized that blue light would induce phosphorylation, and with the wild type enzyme, then you could inactivate, and so that this was a way to deactivate the system after photoactivation. However, we hypothesized that if you had an impaired phosphatase, then you should actually, at steady state, increase the amount of the active phosphorylated PHOT2, and it was clear that phosphorylation was required for normal function. And if that's the case, then we might expect, first, phototropism might be more sensitive, and stomatal aperture at steady state might be, in fact, wider. And so when we looked at phototropism, sure enough, if we got rid of PHOT2, got rid of PHOT1 so that we were just looking at PHOT2 behavior, in the absence of in the impaired activity of the phosphatase, not only did we get a more rapid phototropic response, but we actually got more curvature over time. So the that indicated. We then looked at guard cells. Here they are closed. I won't show you the quantitation, but here's what it looks like when they're normally open. But in the absence of functioning phosphatase with a mutant phosphatase, note that the guard cells are far wider open than they are in the wild type. So in both cases, it appears that the phosphatase is downregulating the functional molecule. Well, it was interesting. The interaction between phototropin 2 affects phototropism and stomatal aperture, but no other PHOT2 mediated response that we tested. they were all absolutely the same with or without that mutation in the phosphatase. And furthermore, PP2A, this same mutant, had no impact whatsoever when what we looked at was phototropin 1 function instead of phototropin 2 function. Well, so our second brush with guard cells, that was our first. Our second brush with guard cells had to do with another of Dongsheng's discoveries from yeast 2 hybrid, and that is that there's a 14,3, the lambda isoform interacts with photoactive phototropin 2 at a particular serine, at that serine 747. And again, yeast 2 hybrid and pull down verified that there was a real interaction. And here you can see in this case that the interaction is now downstream. It's with the kinase domain instead of with the upstream love domains. And here we found that stomatal opening was in fact severely impaired. A couple of experiments. Here it's impaired, except that when we can complement, put the wild type 14,3,3 molecule back in, and we can restore normal stomatal function. And in this case, it was even more highly specific in that this is the only phenomenon, the only phototropin-mediated phenomenon that we could find that was impaired in the 14,3,3 mutant. Highly specific to just that one phosphorylation and association. Well, in 2007, Bill Eisinger joined the lab. He was a long-time professor at Santa Clara University, where it's an undergraduate institution. And I was lucky enough that he wanted to continue doing research, so he joined the lab. And he was interested in guard cells, and guard cells were affected by photoactivating phototropin. So our interests were pretty much in common. So also I had a colleague, Dave Earhart, who had plenty of Arabidopsis that was carrying GFP tubulin. So we decided it would be fun to look at the behavior of the microtubules in guard cells as they closed or opened. And thanks to Dave, we have very, very good confocal microscopy. So this was the initiation of this project. And as I mentioned, there's this very large literature on osmotic regulation of stomatal aperture. People like Mike Blatt, Sally Asman, Julian Schroeder, and a number of others, but only a limited amount of information about the possible role of the cytoskeleton. So, as you know, schemes like this have been developing, and I find them understandable barely, a bit confusing. So here's what Bill first saw. These are guard cells now expressing GFP tubulin, and you see that the tubulin is in hoops around the perimeter of the guard cells. And now this is with the guard cell wide open. If we treat with ABA, which induces closure, a couple of things happen. First, you see there are lots and lots of these microtubules, and it turns out that the brighter ones are probably bundles. I'll come back to that in a minute. But over time, the number of those bundles that you can actually resolve decreases gradually. Here's 30 minutes, 40 minutes. And also, there's been a loss in total fluorescence. And this is interesting because the monomer of tubulin is just as fluorescent, GFP, as is the polymer. So there's a net loss of tubulin, and a rather significant loss of tubulin over time. And then if you correlate stomatal aperture with the number of these things that you can count, you see that there's a pretty good relationship there, pretty clean relationship. And when they close, the number of aperture, the aperture closes, and the time course for the loss in structures that you can resolve goes down simultaneously. And then you can take these plants when they're closed, put them from the light to the dark. They close, the number of elements that you can count goes down. Put them back in the light, they open, and simultaneously up come the number of those bundles that you can see. So why the decline? Well, maybe less growing ends of the microtubules. That's one thing that has happened. Maybe you have the same number of growing ends, but they're growing more slowly. This could account for some of the loss. You could have more rapid degradation at the minus ends, or some combination of those processes. So let's take a look at the growing ends, and let's hope that this works. So we're going to be able to look at the growing ends by looking at a different label, and this is end-binding 1 GFP. This is a protein that's found at the growing ends of microtubules. And let's hope, oops. Yeah. So you see now, this is just watching in time lapse. And these are the growing ends of those microtubules. And having this, you can do two things. First, in the first place, you can count the number of them. And second, you can measure the rate at which they're moving. And so we did that. And this would tell us maybe more specifically. So, moving away from the core? Pardon? Are they all moving away from the core? They're moving in a circular fashion. It's difficult to tell whether they're going under or over. We think that there are some going in both directions, but haven't really studied that in any great detail. But here, if we now look at the number, as the guard cells close following ABA treatment, it induces closing very nicely, but it doesn't change the number of comets, these little comets that you see flying around. So the number of growing ends doesn't change, just showing the pavement cells along with the guard cells. And then if we look at the velocity, closed versus open, there's no difference either. They're going just as fast and there are just as many of them. So we're left with the conclusion that they're actually undergoing differential degradation at their minus ends. Okay, so the question then becomes, well, do microtubules play a major role in stomatal opening and closing? They're appearing when they open, they're disappearing when they close. So do they play a functional role, or is this just simply something passive that happens as a consequence of opening and closing? And there are ways of testing that. There are two drugs, Arizolan that we can use, or I think colchicine does that too, but Arizolan destabilizes microtubules and Taxol stabilizes them. So we can try these drugs and see what effect they have on opening and closing. And in this experiment, we're starting with them initially in the dark. We're putting the leaves into the light. And either Viscia or Arabidopsis, you can see that the guard cells open. On the other hand, if we have treated with Arizolan, these structures don't reform. As you can see, this is just sort of messy inside the cell, and they don't open. On the other hand, if we treat with Taxol, they do open, and they open relatively normally. If we now do the reverse experiment, going from light to dark, normally they would close in the dark. With Arizolan, of course, they do close, and you get this same sort of mess. But in Taxol, they're stuck open. So we conclude, therefore, that the microtubule changes that we observe on stomatal opening and closing, these changes are required for normal stomatal function. One of the questions you might ask is, well, we've transformed these plants and put different stuff into the guard cells than was there naturally. So just maybe we've interfered with transpiration. So this is just a classic water loss experiment done with colleagues in the ecological physiology group at Carnegie. And no matter wild type or any of these transformants, they're all behaving perfectly normally. So we don't need to worry about that. Well, then Bill decided to try to get a little more quantitative. Some of the early pictures looked like that. You could then draw a line and scan across and get a little profile of fluorescence. Here's from the lower cell and here from the top cell. And then you could do some experiments such as this, where when you went into ABA with open guard cells, you notice that the high peaks gradually decline. In fact, they're virtually all gone after whatever that is, 40 minutes. Whereas in the pavement cells, there's no particular change. He then got more sophisticated, or I should say the equipment got more sophisticated, and now we have a better image. And now you can take this and rotate it so that you're now looking at the ends of the microtubules and the fluorescence from the GFP. And then you can draw a line here and do a scan, and you get something that looks like this. And if you sort of squint at this, you could convince yourself that there are some peaks that are down in here, some peaks that are around in here, and some peaks that are here. So maybe this isn't going to be a uniform series from small to large. Maybe there'll be discrete steps. Now, but for this particular one, you don't see that very well. But every once in a while, we'd get something that looked like this. And where one clearly was having a series of increments. And we interpret this as an increase in bundling, as the total number doesn't decline all that much when they close, going down to here, going down to here. But the number of high peaks drops down, and it drops down stepwise. So we interpret that as a decrease in bundling. We're going from singles here to doubles to triples to quadruples and so forth. And this accounts for the difference in the brightness of the fluorescence that you see in those various structures. Well, now, tubulin, as I mentioned, doesn't lose its fluorescence as a monomer. So what are the possibilities? It could be that the loss of fluorescence is likely proteolysis. If that's the case, then if we treat with something that will inhibit proteolysis, just maybe we can block the block stomatal closing and inhibit the fluorescence loss. Likewise, COP1 is an E3 ligase, and the COP1 mutation, COP1 is involved in all kinds of things going on in plants, but it was known that the COP1 mutation prevented stomatal closing. So it might also prevent the fluorescence loss. So here are a couple of experiments that we could do. So we're first thing is, if we inhibit proteolysis with MG132, you can see that I don't have my glasses on, but I assume that that's the stomatal aperture. They do not close. And if you look at fluorescence, the MG132 also very much impairs the fluorescence loss. That essentially reflects photobleaching that occurs under any circumstances. And again, you can see that in DMSO, we go into ABA, we lose the structures, whereas if we have MG132 present, these guard cells don't change particularly in the appearance of the microtubules. Okay. It turns out that a lot more than just proteolysis is involved in closing, because you can inhibit transcription or translation. Here's another protease inhibitor besides MG132. And again, if you inhibit transcription or translation, you prevent the closing reaction, and they remain open in ABA after a 40-minute treatment. So there's a lot going on that at least we hadn't expected. The other thing is, what about COP1? Well, COP1, as I mentioned, doesn't close. Incidentally, if you look at COP1 guard cells, notice that they're more nearly circular than the wild-type guard cells. And note that in COP1, ABA, you can hammer them, and you can't get them to close. So they are essentially stuck open. And not only are they stuck open, but the microtubules are not changed by the ABA treatment. So I guess that's consistent with what we've seen before. Yeah, this shows that's the stomatal aperture unchanged in COP1, and there's this is the fluorescence in COP1 with the controls here, no different. That's just the bleaching, whereas in the wild type COP1 protein, you see the fluorescence loss, again plus ABA, over the same time course. Well, if we disrupt COP1-6 guard cell microtubules with the rhizoline, can we get them to close? And so we tried that experiment, and sure enough, if we treat them with a rhizoline, they do indeed close. And now I've now switched to some work by Roger Khanna, who joined the lab about 3 years ago on a full-time and then a part-time basis, and is still working with us on it. And you notice that if you treat now with a rhizoline, the COP1 guard cells, not only do they lose their microtubules, but they also close. But now, of course, that could be simply maybe we just killed the cells, and with the inhibitor. So what happens if we try to wash it out? Oh, here's just a quantitation. The one to look at is this one. Darker light with a rhizoline in COP1, they close there. So again, did we just kill the cells, or in fact can we recover them? So if we do a washout, the microtubules structures reappear and they open back up again. So we haven't just killed the cells, and again, it's fairly good evidence that these microtubule changes are indeed essential for opening and closing guard cells. Okay, now the question then becomes, does the COP1 mutation in any way affect electrophysiology of the guard cells? Because after all, they do have to get driven by osmotic changes. Potassium has to move in for opening, and all these other things have to go on. And for this, we started a collaboration with Julian Schroeder's lab at UCSD, and Junlin Lee was a postdoc there who did these experiments. And here is the thing I showed you at the very start. And there are two important channels that are worth mentioning. There's this potassium channel, which is essential for opening, and then there's a slow anion channel. Whoops, yeah, there's a slow anion channel, which is right here, which is required for closing. Actually, this is a very much oversimplified diagram. I have another more recent one, but for me, it was just absolutely overwhelming. So I chose the simple one. So required for closing, required for opening. So let's take a look at the behavior of those two channels. And so here's what happened when Junlin looked at the potassium channel, CAT1. In wild type, this is the behavior that you get. It's the usual experiment, voltage, current plot. And you notice that in COP1, it's absolutely normal. There's no impact whatsoever in COP1 on the potassium channel. But to our surprise, when we looked then at SLAC1, the slow anion channel, it was completely dead in COP1. In fact, its behavior in the COP1 mutant was about the same as what you'd expect in a SLAC1 mutant. It was essentially non-functional. Well, that raises the question, what's the interaction between SLAC1 and all these microtubule studies in COP1? And in fact, what is the role of these changes in stomatal function? Structurally, Do they provide some sort of structural contribution? It seems unlikely. They're not all that strong to be able to have a role in changing the shape of the cell. Maybe they're transporting vesicles to and from the plasma membrane. Remember that these cells are expanding out. In our experience, in COP1, for example, where there seems to be no closure, the pair of guard cells forms something that's almost circular, with a circular pore, whereas in wild type, the arrangement is a bit more elliptical, and the pore is smaller. So it's as if there's a balance between opening processes and closing processes that gives you some sort of intermediate state of opening. And when, in the presence of COP1, we kill the closing process, then we're only sealing the consequence of the opening processes, and they are at their most distended. And in fact, they're in an almost circular conformation as a result. When you expand these cells, and we can argue about this later on, we were chatting about shape and so forth, it appears that there's an increase in the outer surface, the size of the area of the guard cells. And this means that one of two things: either if you're going to increase the surface area at all, either you have to make it thinner or you have to add material. And so could it be that the role of these microtubules is to transport vesicles containing proteins and more membrane material for fusion with the plasma membrane, transport to on opening and remove on closing? Well, one of the ways to look at that then would be to look at what is the relationship between these changes and SLAC1. If we could get a SLAC1 fluorescent label, we could follow it. So we started to do that, and these are our most recent experiments. This is SLAC1 GFP, and it's in the COP1 mutant, so that these guys are wide open. And you can see here that the SLAC1, as you would expect of a channel, is pretty much localized to the periphery of the cells, and not much in the interior. Also, if you now look at, and again, these are very preliminary pictures, you see that the arrangement, in the first place, it's near the meridian of the cells, and it's more there than it is on the upper and lower surfaces, the surface towards the air and the surface towards the interior of the leaf. But also, it's showing some kind of structure. And the question is, when they're open like this, does that structure coincide in any way with the microtubules? Because that would at least be a lead that the microtubules could be involved in moving stuff around. Well, what does it look like when they're closed? What it looks like if we now, this is in the COP1-4 mutant, but if we now treat it with a rhizolin to close it, remember we could close it, then we could treat it with washing out and get it to open again. I think that's shown in the next slide. Does the distribution change with stomatal opening and closing? And we're using COP1 as our test system. And you can see that the change is really rather dramatic. When they're open, the SLAC1 GFP is mostly peripheral at the plasma membrane, whereas when they're closed, it now is very much internalized in what appear to be vesicles of some sort, the nature of which, at the moment, we simply don't know. As I say, these are all very recent. Well, so what the next step now, and one which we're just starting, is to get plants that are expressing both a fluorescent SLAC1 and a fluorescent tubulin. But since what we had was, in both cases, GFP, that wasn't going to work. So we've had to work now with different labels. We finally have just gotten some plants that are expressing M-cherry tubulin, which is a red fluorescer, and SLAC1 GFP, which is the green fluorescer. And here's last week's picture. This is the M cherry tubular and the SLAC 1. These are almost closed, and the picture here, of course, is just sort of a mess. But it's the very first picture demonstrating that we do, in fact, have double-labeled, fluorescent-labeled plants, and we're going to be able to take a look and do time course studies and watch and see whether, indeed, the microtubules are involved in moving vesicles around containing SLAC 1 and who knows what else. That's our working hypothesis at this point. And with that, I'll have to say, there's clearly a lot more to do. So I'm looking forward to a lot of neat pictures in the next 6 months. So with that, I mentioned Tong Chung, who did that earlier work looking at post-translational modification of phototropin on photoexcitation. Bill Eisinger, who did the initial work on microtubules, and then Rajneesh, all of this more recent stuff, our collaboration with Julian Schroeder, with special thanks to Junlin for making the electrophysiological measurements. And we're halfway up the mountain, but we still have 600 meters to get to there from here. And that's a different mountain, by the way. And we have a lot more to do, and people are going to be busy. I hope we're going to be busy for a good while longer. And support came from both Carnegie and from the National Science Foundation. And thank you very much for your attention.
SLAC fluorescence or channel in the anticlinal plasma membrane only and not in
It's largely in the anticlinal. We have other pictures of that alone, and there's a sort of a band of it around the anticlinal.
only working with the subsidiaries.
Yeah, I'm guessing we're guessing that it's in fact in contact with it's where it's in contact with the surrounding epidermal cells.
And those comets may be bringing.
The comets could be bringing stuff up to deliver. I think we're in a position that, not with my skills, but with David Earhart's and Rajneesh's, we might be able to probe that and find out whether that's really what's going on. Yeah, the question is whether the hypothesized vesicles that are going back and forth could have some impact on the physical properties of the cell wall. Just my gut feeling is no. I don't mean to reject it out of hand, but I think it's unlikely, because the microtubules start out as being, as the guard cells develop, of course, the microtubules are what determine where the microfibrils are. And that this function that we're hypothesizing is a later one, and it's a function for a mature cell.
You do know that calcium can affect the elastic modulus of the conjugate, and it's a reversible process.
There are ways of testing that now. There are ways of testing the elastic module of a cell wall, and I've seen papers on that. But so it's something we've thought about, but haven't done. It would be very interesting to test just that notion. I think the techniques are out there. Whether we have the skill to use them properly or not remains to be seen. Got to get NSF money for that.
Have you looked at the effect of blue light on the microtubules in the guard zone? Because I think David Earhart's seen blue light affecting microtubule stability in cells in the hypocauple.
That's right. He gets a really elegant reorientation of the microtubules in the hypocautle, which is clearly related to the change in the growth pattern. What we see specifically is that in blue light, the microtubules increase in number and very much increase in bundling. And that's a blue light response. Not other photoreceptors will modulate that response, but without the phototropins, you don't get any light sensitivity at all for stomatal opening. See, it takes about 40 minutes. So there's plenty of time for a lot else to happen. I may have said something misleading when I said the COP1 is involved in the degradation of the microtubules, because it could be involved in the degradation of something else that then plays, it could be much more complicated than that. No, it's purely correlation, and that is that the fluorescence decline parallels the time course for closing, and rather closely. But I mean, these are happening on a time scale of many, many minutes. So the time resolution simply isn't good enough to say more than at this point it looks as if they're closely correlated. Better time resolution might show that that's not necessarily the case. But that's a good question. And it caught me up because I tend to be much too confident when I talk about COP1 and proteolysis and microtubules. It does not have to be that the primary event. It could be indirect.
Can I ask one question? So have you shown polyubiquination of the microtubules, and what triggers the polyubiquination? What's the signal transduction pathway that triggers the polyubiquination?
I don't know about that for the microtubules. I know that there are these ubiquitination studies in a phototropin itself, but no, I can't address that.
That's a prerequisite for degradation by the protease.
Yeah. But we simply haven't looked at that.
It's so trivial, but the peaks you had with the sort of half length and microtubule fluorescence.
Yeah.
What were the widths of those peaks in nanometers or?
Well, what's the length of the guard cell?
Maybe 200 microns.
200 microns. So they could be they could be 1/100th the width of the cell, a micron or so. But they vary a lot. The bundles are going to be bigger than that. The singles. In our earlier experiments, the resolution wasn't good enough to see singles, and that's why we thought we were actually losing a large number of the structures. We're actually only losing a small number, but we're going from multiples to singles.
Thank you very much for a wonderful seminar.
This has been a production of Cornell University, on the web at cornell.edu.